So I wanted to begin by just talking to you about a couple of announcements to your left. The first announcement is that the midterm is going to be a week from this Friday, and it's going to cover material from lectures 1 through lectures 9. The other 10 through 14 will be tested on the final exam. So that'll help you focus your studies, and we will cover those lectures in the review session, which is going to be a week from today in this room from 6 to 7 in the evening. And again, the review session is not webcast, so I would encourage you to come, and if you can't come, have somebody who is going to come here, and they could either record it or take notes. But the review session, and I'll talk about more the midterm and the review session a little later, it's been very useful for students because sometimes the questions addressed in the review session are ones which come up on the exam. So I'll spend more time talking about the midterm probably on Monday, so don't worry about it, and we'll have time to talk about it in your sections and as I come around the lab if any of you need help. Today what I'd like to do is to talk to you about what is arguably the most spectacular process in plants, and the process that most of you think about when you think about plants. That is the process of flowering. Now, the production of flowers by plants represents a tremendous transition in the development of a plant, because at this time, the vegetative pathway, which has been producing leaves and all of the primary tissues, the vegetative pathway ends. So we have the vegetative pathway. It ends. And to remind you of the vegetative shoot axis, we have an apical meristem. And in the vegetative apical meristem, we said that the cells are not committed to any particular developmental pathway. And that in theory, the cells which make up the apical meristem could divide forever. In the production of flowers, or as it is known as the transition to flowering, the shoot apical meristem has its properties dramatically altered. So the shoot apical meristem, which again we'll abbreviate as SAM, is dramatically altered. It's altered developmentally. And the main features that we want to draw your attention to in the alteration of the development is that the shoot apical meristem stops producing leaves. The shoot apical meristem stops making leaves. It enlarges, and it begins to make the floral organs. And to give you a setting for these changes, I'd like to show you the first few slides which illustrate these points with regard to changes in the apical meristem. So this first slide is of a shoot apical meristem similar to what you have seen before. This is in the vegetative state. Here is the apical dome, and here are the leaf primordia, the newest leaves on either side of the shoot apical meristem. As that apical meristem undergoes transition to flowering, it ceases producing leaves, and on its flanks it produces then the various organs which make up the flower. Notice that the top of the apical dome still has not gone complete transition. So that if we now look down on the top, your eye is looking down on the top, the whole apical dome now has been consumed. It has been developmentally reprogrammed to now make all of the floral parts. To give you an idea of the changes in the size of the shoot apical meristem as it goes through this transition to flowering, here is the vegetative apex making leaf primordia, and as the apex is committed more and more to a different developmental pathway, that is to making flowers, in this particular pathway we see that the apex enlarges and that it is consumed by the production of these new floral parts. Now, the studies of flowering and of the controls of flowering, as you might guess, are an important aspect of our lives and also of plant development. And these studies go back to the work of two Americans by the name of Garner and Allard. So what I'd like to do is to discuss with you their work and how it impacted in our understanding of the transition to flowering, that is, in the change of the developmental pathway from vegetative to the flowering pathway. Now, Garner and Allard worked for a government lab in Maryland in the 1920s, and they were interested in breeding tobacco plants. They were interested in getting plants which were very large, which would provide a lot of tobacco for cigarettes. And what they would do in their work is that they would get lots of different seed from all over the world, plant it out into their garden in Maryland, this is where it was located, and then look at the garden and look to see if there are any particular plants which were quite large, quite excessive as far as the number of leaves they were being produced. And then they wanted to collect seed from these plants. And for the first few years after they broadcast the seeds, at the end of the summer, they noticed that there were a couple of plants in their field which were quite large. And they called these plants, they were so large, they called them Maryland mammoth. And they hoped to collect seed from the Maryland mammoth plants. But unfortunately, what happened was these plants had not flowered by the time the first frost came in Maryland. And so the plants were killed, and Garner and Allard were unable to collect seeds from them. So after about 2 or 3 years of this lack of success, what Garner and Allard did is they spied the plants growing in the field in August, and rather than allowing the plants to be killed by the frost again, they dug them up and they brought them into the greenhouse. And in the greenhouse, they were able to show that the plants went on to flower. They were able to produce flowers and seeds. Now, Garner and Allard knew there was something different about the greenhouse environment, and what was different, of course, was that it was preventing the frost from killing the plants. But what was it about the environment which was allowing these plants to flower? Why did the plants not flower until after they were put into the greenhouse? It wasn't simply the fact that they were in the greenhouse that made them flower. And what Garner and Allard discovered, and this is the basic insight to the beginning of flowering, what they discovered is that the length of the day, that is the amount of light which was being produced, we'll explore this statement more in a moment, the amount of light which was being produced gradually got less as the fall and winter were coming on, and they hypothesized that the decrease in the amount of light, or as it is known as the photoperiod, that is the amount of light that the photoperiod, they hypothesized that the amount of light was important in determining whether or not the plant flowered, although they didn't understand how, and that by moving the plants into the greenhouse, these plants then were not killed, but were able then to sense that the day was getting shorter. And to give you an idea of the kinds of day length changes which we see, I want to show you this slide over here. In this particular slide, what we have on the x-axis are the months of the year, and on the y-axis we have the day length or photoperiod. And Maryland is probably equivalent to about Philadelphia, which is about 40 degrees latitude. And what I want you to notice, and your own personal experience will tell you that this is true, is that as the winter and it goes to spring and summer, that the length of the photoperiod increases, and then as fall comes along, the length of the photoperiod begins to decrease. And what they found was that Maryland mammoth would not flower, and we'll write this out for you again, until the length of the photoperiod reached about 11 hours. It had to shorten until about 11 hours for these particular plants. A couple other points I'd like to make on this slide here for you is that on the equator, that is at 0 degrees, the length of the photoperiod is more or less constant at 12 hours. And as you go higher up in the latitude, so that you're in Oslo, Norway, the length of the photoperiod becomes very extreme at the height of summer and very short in the wintertime. So they were suddenly faced with a very interesting issue or conundrum. They now had some indications of the effects which control flowering, but they were meant to be breeding better tobacco plants. And being good biologists, good scientists, what they decided to do was to abandon their work on tobacco, at least on producing tobacco that could be smoked, and to try and understand something about the photoperiodic control of flowering. And so if we take the 24-hour clock and we open it up to make it a straight line, what they found was that if the clock, this is a 24-hour clock, if we open this clock up, that with 11 hours of light and 13 hours of dark, the plants flowered. And that they found that the critical time then, the critical time was 11 hours. If there was more light than 11 hours, the plants wouldn't flower. Very important point. And they call plants requiring some minimum, we'll write this better for you, number of hours to flower, and this minimum cannot be exceeded They call plants like this short day plants. Or as we will abbreviate it, SDP. Having then found that the tobacco plants that they were working with were short day plants, in other words, if you exceeded that critical number of hours of light, the plants wouldn't go on to flower. They then began to look at a wide variety of plants to see what their photoperiod requirements were. And coming from this work was the discovery of a second group of plants. And these plants, if we take our 24-hour clock, again, our 24-hour clock, these plants, let's put this one here, 14 hours of light, 10 hours of dark. These plants flowered, and they only flowered if the critical number of hours of light exceeded some number. And in this particular case, this was the critical number of this particular plant, 14 hours of light. And they call plants which required, in this particular case, more light rather than dark, they call these plants long day plants, or LDP. And finally, a third category of plants were found, and this third category was that the plants flowered irrespective of the length of the photoperiod. And this, of course, always assumes that there's enough light for photosynthesis. And plants which flowered irrespective of the amount of light they got, they call them day-neutral plants. DNPs. This was a profound observation because it allowed people to manipulate the environment in which plants were growing to allow them to have plants flower and to be ready to flower at particular times of the year simply by manipulating the photoperiod. All of you know that chrysanthemums are generally in flower around football season, or Easter lilies are flower at a time when Easter comes, although the date of Easter can change each year. Or this plant over here, a poinsettia plant, is always in flower around Christmas time. Here is the vegetative plant to my left, and here is the plant which has been induced to flower. These red structures are actually leaves, but nevertheless the flowers are also present. All three of these plants, chrysanthemums, lilies, and the poinsettia, are short-day plants. We can manipulate their flowering by giving them photoperiod a certain number of days or weeks before we want them to be in flower. This is an important point and a central point in understanding flowering. Now, Garner and Allard were very excited about these observations because they felt that by demonstrating that it was the length of the photoperiod that now one could be able to control flowering in many plants. And then something strange happened in their observations. Let's look at something which casts doubt on their interpretation that it was the length of the photoperiod which controlled flowering. Let's put our short-day plant up here on the board again. We'll make our 24-hour clock as we had before. And on this short-day plant, we have 11 hours of light and 13 hours of dark. And under these circumstances, this plant, as we said before, flowered. And now what happened was, we'll make a second clock underneath. The plant had 11 hours of light again, but something happened in the dark period. And what happened in the dark period is that somebody came into the greenhouse at night. Maybe the custodian to empty the garbage can, maybe a researcher to water the plants. But they came in the middle of the night, and there was a brief illumination. A brief illumination. And when there was a brief illumination, maybe 5 to 10 minutes, not very long, even though the length of the light period was exactly the same as the above, these plants were no flowering. So how could it be, if the light period is important, if you briefly interrupt the dark period, that you are influencing flowering? They then did a very clever experiment with the long day plants. And this again was occasioned by the fact that they felt maybe they were wrong in their interpretation about the events which go on during the light period. So let's make our 24-hour clock for our long day plant. We'll make our 24-hour clock. We'll put the same thing that we had on before, 14 hours of light and 10 hours of dark. And under these circumstances, we got flowering, just as we talked about before. Now this is the clever experiment they did. They gave the plant 14 hours of light, but then they extended the dark period. They extended the dark period so that the total amount of time which they extended it was 18 hours. So they extended the dark period. And under these circumstances, what they found was that there was no flowering. So coming from this experiment was the conclusion that in a short day plant, flowering is controlled by the length of the uninterrupted dark period. And this again assumes you have enough light for photosynthesis. This is assuming enough light for photosynthesis. If the dark period is shorter than some number of hours, the plant won't flower. So that was for the short day plant. How about for the long day plant? Well, the long day plant, we said, didn't flower if the dark period was extended. So for the long day plant, the dark period must be shorter than some number of hours for the plant to flower. And indeed, people have found that long day plants don't need any dark period to flower. No dark period really needed. So, their original idea that it was the length of the light period which controlled flowering was reversed. We now know that the events which promote flowering occur in darkness, or the events that inhibit flowering occur in darkness for the long day plant. This is an important observation, and it is used and it is central to our understanding of the controls of flowering. So we still use the terms short day plant, thinking that we have a short number of hours of photoperiod, and long day plant, a long number of hours of photoperiod. But in fact, what we really should be pointing out is the period of darkness, the short night plant or the long night plant. This is what we want to have. But the terms short day and long day were first used, and they are the terms that are applied. Now, people have been very interested in studying factors which affect flowering. And Garner and Allard started a big field. We're going to talk about some of the more recent work coming from the flowering experiments. One of the more interesting series of experiments which started as a result of these observations was to ask, are there other environmental factors besides photoperiod which affect flowering? And what Garner and Allard and others have since shown is that in addition to proper photoperiod, many plants require proper temperature. And the temperature that I want to draw our attention to today is cold. So for flowering in many plants, at least for the flowering to be very efficient, not only do the plants have to have a proper photoperiod, but they have to have an exposure to some periods of cold. And what we think happens is that the cold makes the plants more receptive to events triggered by the photoperiod. So let me give you an example of a couple of plants in which the flowering is controlled not only by proper photoperiod, but also by cold temperature. Two plants with which all of you, I think, have some familiarity are celery and beets. These are both short day plants both short day plants, and they require exposure to cold. The effects of cold on promoting flowering are called vernalization. For those of us who live here in the East Bay, or for those of you from Southern California, where we have a variety of plants growing, many of these plants come from the east or from other parts of the world where they normally experience a period of cold. If you want to get these plants to flower, for example, tulips, if you get tulip bulbs, what you are told to do before you plant your tulips is to put them in the refrigerator for about a month or two. What you're doing by putting them in the refrigerator is improving the flowering stimulus, and you're providing a period of cold, which is the vernalization that goes on for the plants. Very important aspect. Here in the Bay Area, we have a lot of plants outside. If you guys go out of the building when you leave, there are some magnolia plants on the lawn. At times when we have a very cold winter and spring comes along, that magnolia plant will burst into flowers as soon as it gets warm. Every one of the buds will open up and flower. In a time of warm winters, as we've had this year, they will not have proper vernalization, and the flowers will come out sporadically throughout the entire year. So vernalization is an important aspect of flowering, an aspect which is important with regard to understanding how flowering is initiated and how it works. Now what I'd like to do is to begin to discuss with you some of the experiments which had to do with regard to understanding the physiology and the biochemistry, and now more recently, the molecular biology of flowering. And I'd like to begin with some of the earliest experiments looking at the physiology. And the first experiment was, what part of the plant perceives the photoperiod? So what was done was to take a plant, put it in a pot, and then what was done was to irradiate different portions of the plant, and it was discovered that the leaf is the site of perception. The site of perception. And it is speculated that the following occurs. The leaf perceives the photoperiod. As a result, a compound was not known at that time. A compound or compounds are manufactured in the leaf. And because we didn't know what the compound was, and because we thought it induced flowering, this mysterious compound was called phloragen. So it's made in the leaf, and then it is transported through the vascular tissue to the shoot apical meristem. Very interesting phenomena. Now, there were a number of many experiments trying to identify what this mysterious phloragen could be. And one of the experiments, which was a pretty interesting one, is the following. If you took a long day plant and you treated with gibberellins, GA, our friend from 2 lectures ago, that you could cause this long day plant to flower. But if you treated a short day plant with GA, there was no flowering. So clearly the GA could not be the universal substance. At least that's what people hypothesized to induce flowering. And to prove this point a little bit further, what was done was there were many, many clever experiments done. And let me tell you one which sort of casts doubt, strong doubt, on GAs being the universal phlorogen. 2 plants were taken. This will be a long day plant. And here we'll have next to it a short day plant. Now, if we enclosed both of these plants in the same environment, let's say it's a long day environment, the only plant that's going to flower is the long day plant. Everybody with me on that? If we enclosed it in short days, the only plant that's going to flower is the short day plant. But remember, GA could induce flowering in the long day plant, but not in the short day plant. So an interesting experiment was done. What was done was to connect the vascular systems of the 2 plants. And this connecting of the vascular systems is called grafting. Some of you may have been exposed to it. And when the vascular systems were connected, we put one plant under inductive conditions. So either the short day plant and when this one plant was put under inductive conditions and grafted onto the second plant next to it, what we found is that both plants flowered. This suggested to people then that the stimulus which was causing flowering was the same in both long day plants and short day plants. Let's look now at a few pictures to illustrate some of these points. First one is a picture of a plant which we have now, in this particular case, enclosed only one leaf. This is a short day plant, and it's put under long days, and we enclose only one leaf under short day conditions, and the plant will go on and flower. Here's our poinsettia plant. These are really leaves. A short day plant, which needs less than some number of hours of darkness, or more than some hours of darkness, or less than some hours of light in order to flower. Here is a plant now in which the plant can be caused to flower by treating it not only with short days, but also with cold. This is a carnation plant where it then flowers. And this is, in this particular case, a long day plant. And in this particular one, you can take this plant and you can actually mimic the effects of the cold treatment on this long day plant by spraying it with GA. You can actually replace the flowering stimulus. Here's a cabbage plant. And in the cabbage plant, again, all the internodes are shortened. This is a short day plant, and when it flowers, its internodes then elongate dramatically. Finally, I want to just show you the picture of this plant right outside the building here, where we have a magnolia plant. And if it is a good cold winter, all of the flowers will come out. This is kind of a halfway winter in which some of the flowers were there and some of them weren't. Here is a winter after we had strong vernalization, and you can see what a difference it makes if all the flowers are vernalized. So these are important aspects of flowering, and we're very kind of up to date on the physiology of flowering. And for a long time, the physiology was all we knew. A lot of people spent a lot of portion of their careers trying to find out what the flowering stimulus was. And in the end, we didn't have a real handle on it until actually quite recently, until about the past 15 or 20 years. And it is now believed that as a result of inducing a plant to flower, as a result of inducing a plant A protein is released from the leaf. We have identified this protein. A protein forms. And moves to the apical meristem and causes gene activation and flowering, or gene reprogramming, if you want to put it that way, and flowering. Now, I'd like to talk to you a little bit more about what happens once this stimulus, which we now think is a protein, arrives at the shoot apex. And much of this work that I'm going to discuss with you very briefly comes from a worker and his group down at Caltech. His name is Elliot Meyerowitz. He's quite a charismatic fellow. Sometimes he comes up to Berkeley and gives talks, and if you ever see him listed as giving a seminar, it's worthwhile. You would understand it. It's worthwhile coming and listening to him talk. Now, Meyerowitz and his crew discovered that once the protein, or they didn't actually know what it was, but once the stimulus arrives at the shoot apex, that genes are turned on. This is not anything surprising in the shoot apex, and that these genes could be classified or broken down into groups depending upon the timing of their turning on. And they had 3 groups of genes. They called them A, B, and C genes that they could group different genes into depending upon when they are expressed or the level of their expression. And what they found, which was the most remarkable part for their work, was that when gene A were turned on, that the shoot apical meristem began to make the sepals. When A and B were turned on, the shoot apical meristem began to make petals. When B and C were turned on, well, let's just say when B is turned on alone, we make the stamens. And when B plus C are turned on, we make the carpels. So they were able to show that the different organs which make up the flower were a response of different types or groups of genes turning on. And from their work, they developed the so-called ABC model of flowering. And that model, as a cartoon to help you remember it, has B here, A here, and C here. If only A is turned on, we make sepals. If A and B are turned on, we make petals. If B and C are turned on, we make stamens. And if only C is turned on, we make carpels. So these observations went a long way in helping us to understand the controls of flowering. Very important observations. They give us a setting now for the work in understanding some of the molecular biology of flowering. This is an area of intensive investigation, an important area of investigation, because if you can control flowering, if you can control when plants produce flowers, economically you can control whether when we get fruits or when we get seeds. Yes?
I'm kind of confused by your diagram.
Pardon me?
I'm confused by your diagram you just drew because you said previously that these stamens.
Only if B alone is expressed as stamens.
Oh, okay.
Right.
That diagram, I kind of.
Yeah, just B alone, just like C alone is so only B alone.
Oh, okay.
Okay? Oh, B and C alone. Oh, I'm sorry. No, this is right. This is correct. You got me on this. B plus C, and then this would be just C. Good. Now we're in shape. Thank you for pointing that out. Right. So only C alone would be carpels. B and C together would be stamens. Thanks for making that change. Everybody clear? Okay. So this has been an important model, is now being used and refined, and many of the workers now base their approaches looking at flowering based upon this model. Now, obviously, if we're going to get flowers produced, the product of the flowers, aside from the beauty, what we really want are the fruits and the seeds. And what I'd like to talk about in the very end is the production of fruits and seeds and something about the physiology and biochemistry of fruit and seed production. And in order for us to appreciate what I want to discuss, I want to go back to a question that a number of you have raised at various times, either when I've been in the lab or in lecture. And I want to go back to the strawberry. We want to talk about fruit production. What we said when we studied fruits in the lab was that in general, the fruit is the product of an enlarged ovary. But we also hinted to the fact that there are some fruits, or things that we call fruits, which represent more than an ovary. And the structure that a number of you have talked about with me is what we call a fruit, but represents more than an ovary. And what I want to do is talk about the structure, which is the strawberry. So this structure up here in front that we think of as a fruit actually represents more than an enlarged ovary. If you look carefully at a strawberry, the things that get stuck in your teeth, each of these things that get stuck in the teeth are a single seeded fruit. A single seeded fruit. And they decorate this structure, which is known as a receptacle. So let's just put this aside now for a moment, and we will return to it when we talk about another aspect of the experimental approach to fruit development. Now, the first thing which individuals noticed when they began to study fruits is they were interested in understanding something about the shape and the size of the fruit. And one of the first observations which were made was that when you tried to understand something about the shape of the fruit, what you found is that the placement of the seeds was in the region where the fruit was largest, in this particular case, the pear. And in fruits such as this squash, your experience will tell you that the seeds are placed down at the bottom. And what was found is that the larger the number of seeds, the larger the fruit, and if the seeds are displaced proportionally to one part of the fruit than the other, that part of the fruit grows bigger, as contrasted with something like the spaghetti squash, which again, for those of you who have cut it open, you know that the seeds are fairly uniformly distributed throughout the spaghetti squash. So what is it about the seeds, if there is any correlation between the seeds and the particular fruit? And what was found when people analyzed the seeds was that the seeds turned out to be the site of production of a large number of growth regulators. But one growth regulator in particular attracted people's attention. So the seeds produced growth regulators. And the one which attracted people's attention most was auxin. It was hypothesized that somehow the auxin was actually controlling both the shape of the fruit and its size. But how would you test that? What kind of experiment would you do? If you wanted to test that experiment with something like a pear, you'd have to dig into the pear and take out the seeds. Or in this squash, you'd have to injure the squash to dig into it to take out the seeds. Not a very good experimental system. So this takes us back to our strawberry. In the strawberry, the seeds are on the outside of this particular structure. And so what was done was to take receptacles before they expanded, before they got very large, and to remove all of the single-seeded fruits. They removed them all. And when all those single-seeded fruits, which were a site of auxin production, were removed, the receptacle didn't enlarge. But if you took that same receptacle from which you had removed the seeds and you sprayed it with auxin, it got big again. So the view today is that much of the physiology of fruits, both their size and their shape, is due to auxin, which is being produced by the seeds. Now, one of the aspects of seeds and auxin production is the fact that there are some fruits which don't have seeds on them. And this is of interest of us because if you live here in the East Bay and you try to grow tomatoes or green peppers or eggplant during the summertime, it's very difficult to grow these crops because the temperature is generally too cool for the pollen to develop. But because auxin has been shown to cause fruits to enlarge, you can actually spray it on the flowers, and this is usually done by home gardeners, and you can cause the flowers to expand their ovary without pollination, without fertilization, and you can get a fruit. This is a seedless fruit. So for those of you who have experienced growing tomatoes here and spraying them, you have no seeds inside. We have also seedless watermelons. There are seedless grapes. There are a large number of seedless fruits which you can buy. Are they all produced by spraying the plants with auxin? Probably not. Those plants probably have fertilization going on, and then during development, the seeds abort. A good example of that is a banana. If you guys eat a banana, the little teeny dots that are inside the banana are aborted seeds. This particular plant, as many other plants, cultivar-wise, aborts its seed. Perhaps the one that has been cultivated most in human history are figs. There are cultivars of figs in which the seeds abort, and the abortion of the seeds then makes a seedless fruit. So this is an example of use of auxin to make seedless fruits, but the development of seedless fruits is a naturally occurring event in nature. You might ask, how do these plants then grow? How does a banana grow if it can't make seeds? And the answer is that bananas are propagated vegetatively. You just take a shoot off of it and you put it in the ground. The danger in that sort of approach is all of the bananas in a particular area, for example in Guatemala or Nicaragua, wherever they come from, are all of the same cultivar. And so if a disease comes through, it's going to wipe out the entire banana crop. As you will see from the slides that I'm going to show you now in a moment, in fact, many fruits like banana actually can make seeds in their natural environment. Let me show you some pictures now of seed and fruit physiology with regard especially to auxin. Okay, as soon as this goes on here, sorry. This first picture is of a squash similar to the one I have up here in front. And again, what I want to point out to you is that the seeds are placed down in the area of the squash which is largest, and this is presumably a result of a higher concentration of auxin, giving you this sort of swollen appearance of the squash. Here's a banana. If any of you have gone to the tropics or gone out to the tropics, you can find different types of bananas growing. This is the banana cultivar in which seeds develop. Here is a banana cultivar similar but much smaller than the one we eat, in which the seeds abort. So this banana essentially would be seedless. This one would not be very much fun to eat. Here's our strawberry. Here is the receptacle when it is very tiny, and after fertilization occurs, each of these structures develops into a single-seeded fruit, making auxin, causing the receptacle to enlarge. And here are the experiments which were done. If the seedless fruits, the single-seeded fruits are taken off, you can cause the strawberry to develop any shape you want. If you take them all off when the receptacle is small, you get a very tiny strawberry, but if you spray the strawberry with auxin, you can get it to enlarge into a strawberry without seeds on it. Finally, I wanted to show you these fruits which are being marketed by Whole Foods. Many of the customers in Whole Foods complained that they were difficult to pick up the grapefruits because they were round, and so what was done was to make finger holes in these grapefruits. And so people can pick up the grapefruits now more easily. I'm just kidding you, but this is a challenge. But this is the kind of thing that you can do with fruit and seed physiology. Okay, I'll see you on Friday then.